How to sharpen memory during old age. The billions of neurons in the brain present at birth never multiply except in the hippocampus. Hippocampus is the seat of memory and buried deep underneath the folds of the cerebral cortex. The hippocampus allows us to form new memories of what, when and where that help to navigate our lives. Neurogenesis, the generation of new neurons from stem cells, is critical for keeping similar memories separate. Neural stem cells in the hippocampus continue to generate new neurons. This incite a struggle between new and old as the new attempts to gain a foothold in memory forming center of the brain. In a study published in the journal Neuron on August 9, 2016, Harvard Stem Cell Institute researchers at Massachusetts General Hospital and the Broad Institute of Harvard and MIT, in collaboration with an international team of scientists, found they could bias the competition in favor of the newly generated neurons. Mr. Sahay, Ph.D. is the principal faculty member and the study's corresponding author. As the human brain matures, the connections between older neurons become stronger, more numerous, and more intertwined. This makes integration for the newly formed neurons more difficult. By the time, neural stem cells also become less productive, leading to a decline in neurogenesis. With fewer new neurons to help sort memories, the aging brain can become less efficient at keeping separate and faithfully retrieving memories. The research team selectively overexpressed a transcription factor, KLF9, only in older neurons. This eliminated more than 1/5 of their dendritic spines, increased the number of new neurons that integrated into the hippocampus circuitry by twofold, and activated neural stem cells. When the researchers returned the expression of KLF9 back to normal, the old dendritic spines reformed, restoring competition. However, the previously integrated neurons remained. Because we can do this reversibly, at any point in the life, we can rejuvenate the hippocampus with extra, new, encoding units. The authors employed a complementary strategy in which they deleted a protein important for dendritic spines, RAC1, only in the old neurons, and achieved a similar outcome, increasing the survival of the new neurons. In order to keep two similar memories separate, the hippocampus activates two different populations of neurons to encode each memory in a process called pattern separation. When there is overlap between these two populations, it is more difficult for an individual to distinguish between two similar memories formed in two different contexts. If the memories are encoded in overlapping populations of neurons, the hippocampus may inappropriately retrieve either. If the memories are encoded in non-overlapping populations of neurons, the hippocampus stores them separately and retrieves them only when appropriate. An increased neurogenesis had less overlap between the two populations of neurons and had more precise and stronger memories, which demonstrates improved pattern separation. So, an increased neurogenesis in middle age and aging exhibited better memory precision. By increasing the hippocampus's ability to do what it's supposed to do and not retrieve past experiences when it shouldn't, can help. This may be particularly useful for individuals suffering from post-traumatic stress disorder, mild cognitive impairment, or age-related memory loss. Thank you.